So those are my kids, and like many of us, they really like to go out with me to a coffee place, drink orange juice, eat nice croissant or chocolate cake like we just did in the coffee break. And you know who worked really hard after that nice feast? The pancreas. So the pancreas is a small organ that's found behind our stomach. It looked like a tree. It composed of a network of ducts. And in between those ducts, we have these islets of cells. The pancreas has two functions, blood sugar regulation and digestion. The islets, the cells that produce hormone, the famous one of those are its insulin. It's only 5% of the pancreas. The rest of the pancreas are part of the digestive system. Those are the cells that produce the digestive enzymes. We call the islet of cells, we call this the endocrine compartment of the pancreas, and the digestive part we call the exocrine compartment of the pancreas. So why study pancreas? So over 400 million of people suffer from diabetes. It's a disease that happens in the endocrine compartment of the pancreas. There is the pancreatic cancer. Unfortunately, it's one of the deadliest cancer forms that happens in the exocrine compartment of the pancreas. So in my postdoc, most of the researchers really focus either on the endocrine compartment or the exocrine compartment. In my postdoc, we wanted to build a tissue-like pancreas in the lab that will be formed from both of these compartments, both the endocrine part and the exocrine part. We wanted to do it in the lab in a dish because we wanted to better understand pancreas development. We wanted to get insight about pancreas syndrome. We can manufacture this structure that might mimic the architecture and function of the pancreas in high numbers. Then we can model diseases like diabetes or cancer. We can do high throughput drug screening, and as many research aspired, and in the future we can use it for cell therapy. For example, producing cells that will sense the glucose and produce insulin. So this is challenging, really building a pancreas in the lab. It is challenging. We need to be able to tackle this challenge, we need loads of cells. So we decided to work with pluripotent stem cells, with human embryonic stem cells, or we started with mouse embryonic stem cells. These cells can become any part of our cells in our body, right? So our work was first to take this pluripotent stem cell that can become to the different cell types and kind of guide it to become the identity that we wanted. For example, we don't want it to become intestine, but we wanted to make them follow this path and become with a pancreatic identity. Once we get the cell that has this pancreatic identity using specific nutrients and molecules, then we can produce these cells in high numbers. And this is me with Abby, a PhD student in the lab that worked with me on the bioprinted project. And this is, I would like to thank her that she worked with me. And we're standing next to the bioprinter, which I will explain just in the next slide. So we have these pancreatic cells. Now we wanted to build this pancreatic tissue, right? So we decided to use two techniques. The first one, we call it the organoid culture, aggregating the cells. And the second one, the bioprinting. So in the first method, basically, we are taking these pancreatic cells, we're mixing them with support cells, and we let the cells sink in the U-bottom well and let them self-organize and find themselves. In the second part, we print the cell in specific location. So let's start with the first method. These are how the aggregates look one day after aggregation. We can see the cells find each other, right, proliferating the well. And what we found that basically after 14 days in culture, when we grow those cells, these aggregates in the lab, they could differentiate, they could become the different cell type of the pancreas, both the cells that produce the hormone like insulin and the exocrine part, the cells that produce the enzyme, and the ductal cells. This is an example of what we got. We can see here the cells that will become a tube cell, a duct cells of the pancreas. And here we could see that in our aggregates we could find cells, endothelial cells, that will build the blood vessels of the aggregates. So moving on to the 3D bioprinting. In this technique, we take cells, we mix them with a hydrogel, with biomaterial, which we called bioink. We designed, this is our bioprinter. We are, as an engineer, can design whatever we want to print. We can use different biomaterials with a printed head, different cell type, and then we can print, for example, here we printed the Technion Lego in a support bath. After we finish this printing, right, we dissolve this printing bath and we get only the printed construct. Utilizing this technology, we can ask different questions. For example, how can we affect the cellular identity of the printed construct using different biomaterial? Using different cell interaction, how cell type A influence cell type B? We can ask how the geometry really influenced the identity of the cells, right? So just to share with you a few interesting results that we got during this project is, for example, we wanted to check how endothelial cells here in red, the cells that will become the blood vessel, will influence the pancreatic cells here in green. And what we found that the pancreatic cells that were in proximity to these blood vessels become more endocrine cells, more cells that produce hormones versus the cells that are found far away from this compartment. Another interesting thing that we found is that when we changed the biomaterial, the hydrogel we used, when we printed our pancreatic cells in a soft hydrogel, what we saw that our construct kind of shrunk over time, versus when we change the platform, when we change it to a stiffer one, right, we can maintain the construct shape. And an interesting thing that we found that on this stiff platform, this pancreatic cell became more endocrine than exocrine. So they produce more hormones, more insulin than the rest. So just to summarize what I just told you, so first we took the pluripotent stem cells and we established a really efficient differentiation protocol. We took this pluripotent stem cell and kind of narrow its path and make it become pancreatic cells. Then we used two techniques of a 3D culture, what we call, either using the aggregation protocol or using the 3D bioprinting. In both techniques, we observed the two cell types, the two compartments of the pancreas, both the exocrine and the endocrine. Using 3D bioprinting, we can ask and get answer of different, for example, asking how the different cell type influence the identity of the pancreatic cells, how the material influence the identity of the cells. With that, I would like to end and thank Shlomit Edri, my postdoc mentor, the lab members, our colleague Francesca and her lab member, Christoph and his lab member, and of course you for listening, and I'll be happy to take any questions.